as I've said before, the toxins in the venom arsenal can target several molecular targets, whether they are G-coupled receptors, nicotinic receptors, whether they are tyrosine kinase receptors. There are various kinds that are found on the membrane of a cell. And these, for the most part, most of the toxins in, especially our killer snails, are membrane-acting toxins. They don't necessarily have to penetrate into the cell, but they act on receptors that are found on the membrane of cells. And so here is an example of how the toxins from a snake, for example, when they work on blood. These are very hemotoxic. What that means is that you have these snake metalloproteases, which will then poke holes into the membranes of the cell and then cause a rupture. And this is how the blood then is released from the artery, and then it starts to clot because there's so much blood everywhere. So you get basically a hemorrhage. And in this one, here's again how the toxins from our cone snails work. The neuron postsynaptic junction, in which the signal, this is specifically how M7A, the pre-alt peptide, works. So it blocks something called N-type calcium channels, and it only blocks those calcium channels. It doesn't block another kind of calcium channel, so that when you're feeling pain, or when it suppresses the pain from the N-type calcium channel, it doesn't suppress pain from like putting your hand in a fire. But basically, this peptide then blocks this N-type calcium channel, and it prevents the release of neurotransmitters, so then the pain signal isn't propagated up your spinal cord to your brain. So to summarize how these toxins work on cell membranes, you have small molecules, peptides, and proteins, and all of them work again at the surface of the cell membrane. And for the small molecules, you have examples like cytotoxins. For the peptides, you have the example that I gave you of ziconotide or M7A, which blocks a specific calcium channel. And then in the proteins, you have things like the metalloproteases and the phospholipases, which attack and rupture the membrane itself. Let's now talk a little bit about the physiological effects of venom. So when we say something is attacking the nervous system, and we're calling it a neurotoxin, that means it's preventing cells in the brain from receiving signals and working exactly as it should. When we talk about something being hemotoxic, it's acting in the circulatory system. That means how your blood is getting through your body, and it's causing these blood cells to burst or to rupture. And when we talk about something being myotoxins, or means it's working in the muscular system, and it's causing these muscular systems to not connect and function properly, right? And so these physiological effects are what is so lethal about venom, but it's also what could be potentially so beneficial when we're talking about venom for medicinal purposes, because we can hone in on which effect that we want to either amplify or suppress according to the disease or disorder that we're treating. And so here is an example of the different physiological effects of snake venom. And so 19 different, these are venom snake toxins, and then that's what VST stands for. And then AVP is accessory venom proteins. Together, this cocktail of venom is what's giving you the different physiological effects that's shown here. So whether it's hemorrhaging, whether it's something that's dealing with hypertension, whether it's something that's leading to muscle tissue damage, all of these things are found within snake venom. And this is how they're impacting the predator, prey, or human when they're injected into that organism. Here's an example of our work in which we're characterizing how different the toxins from different snake venom and what their potential physiological effects are. And so these seem to have hypoglycemic and muscular effects when they are targeting different things within the body. And they also, like we said before, the majority of them are neurotoxins, which are shown in the green bar on the left. All the way on the left are the 3 different types of snails that we characterized. We took their transcriptomes. And in the middle is what we found in those transcriptomes. So what were the peptides in the arsenal being expressed, or the potential for them being expressed? And then all the way in the end is the physiological effect of those peptides. And so you see that a large part of these peptides are mostly neurotoxins, but you have also things that are acting at the muscle level and things that are acting at the hemoglycemic level as well. So for this last slide, when we talk about sea anemone physiological effects, you'll notice that it's similar to what we've discussed already with the snails and the snakes. And that's what we talk about having venom being a convergent trait. So a lot of the compounds that are found in the arsenals of snakes and snails are also found in sea anemones, and they have similar physiological effects, meaning that you have also things that cause hemotoxic effects in sea anemones and things that also cause neurological effects as neurotoxins in sea anemones, similar to what we've seen before.